So thank you very much. Thank you for the invitation. Thank you for the good presentation. And I hope I can bring you information on what is available nowadays. Just to give you a short background, you will see the slides have been translated into Italian, but unfortunately my Italian is not good enough. It would hurt your ears, so I hope you apologize my use of English for this presentation, and I hope that it will still be understandable. Just to give you a very brief background on myself or what I'm talking about, there is really a scientific development on alternative methods. I myself have a chair for development of alternative methods, and we have a whole center at the University of Costanza together with Johns Hopkins University, which is our partner in a joint venture. I'm not only an academic researcher who might not know what is going on in real life. I've spent many years in industry, developing drugs, doing animal experiments as well, and knowing benefits and disadvantages of different technologies. So this is why I was just putting in a little bit of this background information. And I think the most important background information is that we are actually experiencing a silent revolution a lot of the population does not know anything about. So we do not talk science fiction. I do not make predictions like the speaker before. I talk about things that are available today and that have happened. So industry has started changing technologies. Animal experiments have been developed 60 years ago, and sometimes there are much better technologies. We wouldn't use a telephone that is 60 years old. We like to use different technology. And in many areas, animals that have been used before have been substituted, and there has been a huge reduction of number of animals being used for drug development. Sometimes 9 out of 10 animals have been reduced, and definitely in all publications, in all scientific fields, be it neuroscience, be it cancer, be it others, there is less use of animals per publication than there was 20 years ago. And the drivers were not only ethical reasons. As we have to admit honestly, and as you're probably aware of, this has been because the new technologies have something to offer. Lower costs, higher speed, and better information. And this is why these things have been happening. So industry is always fast in adopting the best method available. And now we are in a second phase of what is changing. And this is because the European Union, for instance, has invested a lot of money in development of alternative methods that have been thoroughly validated. So they have been compared to animal experiments, and they're only allowed legally if they're at least as good as the animal experiment. Sometimes they turned out to be better. And industry is, of course, interested in fast data acquisition. So we now have technologies available with the use of robotics that can handle artificial tissues to generate 10,000 data points, where before perhaps 10 animals could be used per day. And these are the drivers for the next phase, and I'm going to show you perhaps more illustrative, more colorful pictures of what is actually available today. So just as introductory experiments, we can look at some examples where there has been use of animal technology and use of non-animal technology. And the most famous example, and I apologize for the pictures which are not really appealing, but this is one of the best known drug catastrophes we had in Europe. That's a drug that was sold under the name of Contergan in Germany. It has the ingredient thalidomide, and it caused the malformation of limbs. And that was tested in animals. But mice do not react to this drug in this way, and animals also don't, apart from one specific type of white New Zealand rabbit, which has not been used in these testings. So these things could have been predicted, for instance, by the use of stem cells, as is a common technology nowadays. There is often the prejudice that we might use animals for some simple applications, but it's very difficult for systemic toxicity or cancer or complicated areas. And I want to present you the example of the drug Ticinero, from the company Ticinero. It was called TGN 1412. This drug was tested not only in mice, rats, and rabbits, but also in monkeys. And it was found to be safe. It was a systemic modulator of the immune response. And then it was tested in humans, and 5 nearly died immediately, a few hours after application of the drug. Big surprise. So this was examined by the British health authorities, because this trial was started in Britain, and they came out with a publication afterwards. And this is the English text. And you see a lot of these people that are involved in the regulation of these drugs in the UK. And the central statement from the abstract is written below. They state that these methods, which they use in vitro now, would have been able to predict the toxicity in humans, and they are going to use such methods in the future. So simple cells would have told the catastrophe where the monkey experiments did not. And this is not opinion, so I'm trying to put in information by reliable sources here. Another question is, how about cancer? And there we can ask the question, if we test 200 chemicals in mice and rats and rabbits and guinea pigs, how well do the data agree between two of these species? Because usually the extrapolation to humans, the prediction for humans cannot be better than between two of the animal species. And if we look, for instance, if we test 100 drugs in rats and in mice, only 57 will show the same result. So how can the prediction from mouse or rat to human be better? This is the problem we are being faced with when we rely too much on animals, as we have seen in the talk before. And there's reliable data on these shortcomings of these assays. And if you perhaps just focus on the last point, number 5, the Environmental Protection Agency, that's the US Agency for Environmental Chemicals, does not even accept these data if they are positive. So 58% of the data where cancer has been found in the mouse has not resulted in a regulation because these data are so unreliable. This is hard background data. So how about other areas? Sometimes it's very difficult to know the human situation. There is a possibility to know it, for instance, for acute toxicity because of suicide cases. So people have had chemical accidents or suicides, and we can now compare how do cells and animals and humans compare in these cases. And we see if we take very simple cells, so these are just skin cells, and compare them with animals, there is not a very nice correlation. The question is, which of these is right, is hitting the center where humans should be? And since we have some human data on these chemicals, and this is a study that has been performed not by some university, not by a company, but by a collaboration of the American and the European Union regulatory agencies, ECVAM and ICVAM. And they found the correlation of the animal with the human data is 56%, and the cells seem to do better, at least they certainly do not do worse, and these are very simple primitive cells. So model systems never reflect reality perfectly, but the cells can do at least as well as the animals. Another big area where we have data from humans and from animals and from a cell model is pyrogenicity. And this is an area that is difficult to understand, but it is much larger than it is assumed. In this area, 200,000 rabbits are being used per year per testing, because every injection drug has to be tested whether it triggers a general fever reaction in humans that can be lethal. This is called septic shock, and it's still the major cause of death on all intensive stations in Italy and also in Germany. So drugs have to be tested for this, and the classical test is shown here. This is the rabbit pyrogen test, and the idea was developed during the last 10 or 15 years that one could simply take human blood, and this can even be frozen blood. It's very easy to transport, and to do a very short assay that can be done in 3 hours and measure a fever mediator. These tests have in fact been developed in part at our university, in part in other places, and they have been validated by the European Validation Agency, which is situated in Ispra, Italy. It's called ECVAM. And this was a study which was run all over Europe in 10 different laboratories and independently evaluated by statisticians. And they found out that this human blood-based, very simple assay was having a higher sensitivity, a higher specificity. It was more accurate in the prediction, it was more reproducible, and it delivered in general better data. So these 200,000 rabbits per year were used for nothing very useful compared to what one can obtain now with these tests, and they have been legally implemented during the last 5 years in all European countries and also in the United States. So now, after having started you just in seeing that there are some areas that are not science fiction, but things have happened already and have gone into legislation, we see what is under development. And again, it's not my personal opinion. This is, for instance, the famous magazine Science, which was bringing out this article last year, that sometimes the in vitro methods may in fact be better in predicting toxicology. And this is backed by people that are really well established in science. Francis Collins is the director of the National Institute of Health in the United States, and before he used to be the leader of the Human Genome Project. And he has formed an alliance of all big American agencies. They produced also in the magazine Science an article on this, and he stated that he is proposing a shift from animal experiments to new technologies which are based on in vitro assays or assays using lower organisms like flies or worms and computational modeling. And this little article of 2 pages has transformed the whole health policy in the US. They're investing major amounts of money into this new development. This is actually the memorandum of understanding which was signed by all these very high-standing people of the national agencies responsible for all the safety of drugs, chemicals, chemical-related substances in the US on moving, and that's why you see a robot in the picture, on this high-throughput screening-based, very modern technologies that generate high amounts of data every day. And just to show you a little bit how this looks like, I've brought along some examples, and this is the low tech. So this is the classical approach where one can do a one-to-one substitution, the classical testing of a shampoo, whether it burns your eyes, whether it's toxic for the eye, used to be the Dre's eye test. And this has been substituted by a simple, clever idea of using the eyes from cows from a slaughterhouse and simply testing with these eyes. This has been validated in over 10 years in dozens of laboratories, and it has shown that it gives the same data, and this is the test that's being used nowadays. Now, the next step would be not to take some material out of an animal, but saying we would rather like to have human material. And some companies got the idea with the first simple material, this is human skin. And one of these companies is called Skin Ethic. It was bought recently by L'Oreal. L'Oreal bought a second of these companies. They make profit, they make black numbers. So these companies are running well in producing human skin, which can be used for testing of cosmetics, but of course also of other toxicants and drugs that might cause an allergy, of doing research in melanoma, all kind of things that are much more usefully done on human tissue than on a mouse tissue. So this is one of the showcases of the development of tissues made from human cells, and this is a development that goes on with much more complex tissues than skin. And this is called nowadays, by a strange word perhaps, human on a chip. This is because these tissues are created as very small tissues. And for instance, the US Ministry of Defense has realized a lot of things they would like to test cannot be tested on humans. There are no suitable animal models they would trust in. So they said, we will invest all our money into generation of such tissues, combination of these tissues, and just in the last year alone, they have invested 200 million to the Wyss Institute in Boston to create such tissues. And to give you a little impression how this looks like, so the miniature format is shown on the right. This is actually a small chip, and this is a lung tissue that can breathe. So on a flexible membrane, you can have different types of lung cells, which are superfused with air, and they are perfused with blood, and it can move and show the mechanical forces, and this is what is being used for study of lung infections nowadays already. So this is a combination of high engineering technology with biological technology, and some of these cells which are not easily available are being produced from human stem cells. And the lung was only an example, so of course there's parallel development of liver and kidney, and then as you see to the right, you might combine these different organs by an artificial blood circulation, and then also see how different organs interact with one another. And again, we are not talking science fiction. This is going on today already in many places. So this is organs. Another area we are interested in, and Dr. Rice has addressed the problem of development, and for instance, development of autism, and we would like to understand how chemicals interact with development of an embryo. So the US EPA has started a huge project which is called virtual embryo. This is basically taking all the chemical information we have about regulation of cells, putting them into supercomputers, and see whether this can be modeled. In Germany, we have a similar project on the liver, which is called the virtual liver. And unfortunately, this is not running now. It ran before. So you would be able to see on the video how such a model can look like, how different chemical gradients can be seen in a developing limb tissue. And these types of models have already been used to study the toxicity of thalidomide, this drug which caused these limb malformations, and it can very nicely reproduce not only the effect itself, but the right concentration at which the effect occurs. And this can also be used for other developmental problems, like a cleft palate, which is a very special word, and I think in Italian it's called labro leprotino, so this cleavage of the palate. Also this can be nicely modeled, and this is not so infrequent in the human population. So as an example to show you how high throughput data are being used, I'm just showing a brief slide on the American ToxCast program. This was again presented by Francis Collins, who I presented before, and he had in his article also a nice overview of the speed of data generation. If we use a mouse, how many data we could get for complicated questions like cancer, and how many data we can get per day or per second if we use these high throughput robotic screens. And the Americans thought about it and then also put the money into it and did it. So they put together 600 different assays for chemical effects, so different biochemical effects that can change a cell, and tested for all of them. And they did that for over 1,000 substances, which they know they are frequently found in our environment, in food, in pesticides, other things. And so they started gaining experience on this, and this was possible because of large robotics units. And now they have expanded this to 10,000 compounds, and this is a screen which is going on and should be finished in 2014. And then we will learn how we can use this biochemical knowledge, which we have gained in the last 20, 30 years together with very fast robots to test it, to predict what chemicals are doing on real human cells and human tissues. And this is, just to give you a break, some easier pictures, no numbers on them, but two people that have provided a very important technology, because you might still ask, how do we get human brain tissue? Nobody likes to donate brain. So these people have found a way to produce stem cells which enable us basically to generate each of the 240 different cell types we know in humans. They got the Nobel Prize for this, and the stem cell development is actually explosively developing, and this is a research that has substituted animals in many areas. It's being used, of course, for pharmacological research. One can study the development of organs right from the beginning. One can generate disease models that would not exist in a mouse. One can do toxicological studies, and of course, also regenerative medicine is possible. This used to be one of the original ideas, but you see there's many other applications of stem cells. And just to remind you, these are so-called induced pluripotent stem cells. So they do not require the use of embryonic stem cells. These cells can be produced from skin, from anyone, no matter whether you're 18 years old or 80 years old. They can be produced from one hair and even from urine. Animals are not possible, and we get human cells with the normal human genetic variation. And of course there are big investments. The European Union IMI project is running already. It's a 54 million project to generate a bank of these cells, and a new banking project is being started today. There have been 60 million involved in other European projects to promote this research. And of course, this is a good investment and good money, but compared with the Americans that sometimes get more enthusiastic about new technologies and have invested $800 million in California alone to transform that state into a stem cell-based research unit. And this has helped already, so there is already new results on Parkinson's and Alzheimer's disease that have not been possible from animal experiments. And so I'm finishing for those that still are perhaps skeptic, they have questions in mind. Some things may not be addressed, of course, with a cell in a culture dish. Of course, you might name diseases like migraine or autism or drug dependence. How could the cells tell us anything about this? But then I just leave the question standing, what would the animals tell us about this? Do we have autism in a rat? Do we have Alzheimer's in a rat? Can a rat have migraine on the weekend? So we have different technologies, and we would be stupid not to use the best that is available today. Thanks a lot.